Every living organism is made of cells. This forms the basis of the cell theory, which states that cells are the basic unit of life and arise from pre-existing cells. The animal cell is a eukaryotic cell with a true membrane-bound nucleus, typically ranging from 10 to 100 micrometers in size. Unlike plant cells, it lacks a cell wall, chloroplast, and a large central vacuole. Instead, it contains centrioles, lysosomes, and has the ability to change shape, making it a dynamic unit of life. Now let's take a closer look at the cell membrane. We are viewing it edge-on in cross-section. Above is the extracellular fluid, and below lies the cytoplasm. You can rotate to explore and zoom in to see the structure in detail. Phosphate heads, shown as blue spheres, are hydrophilic, meaning water-loving. They naturally orient themselves toward water, lining the outer surface of the membrane where they face the extracellular fluid. Each phospholipid has two fatty acid chains, shown in yellow. These tails are hydrophobic, meaning water-fearing, and they point inward, away from both the extracellular fluid and the cytoplasm. A second mirror image layer forms beneath the first. Its fatty acid tails point upward, meeting the outer tails, while its heads face the cytoplasm. Together, these two layers form the complete phospholipid bilayer. Cholesterol molecules, shown in lavender, fit between the phospholipids like spacers. They help buffer membrane fluidity, preventing the membrane from becoming too rigid in low temperatures and too fluid in high temperatures. This stabilizing component is found only in animal cell membranes. These protein complexes, shown in pink, span the entire phospholipid bilayer, reaching both the outer and inner surfaces at the same time. They function as ion channels, carrier proteins, receptors, and pumps, controlling what enters and leaves the cell, while also enabling communication with the environment. Peripheral proteins, shown in green, are loosely attached to the surface of the membrane. Unlike integral proteins, they do not pass through the bilayer. Instead, they sit on either the inner or outer surface. They function as enzymes, cytoskeletal anchors, and signaling molecules, helping the cell maintain structure and communicate internally. Carbohydrate chains, shown in orange, extend from proteins on the outer surface of the membrane, forming a protective layer called glycocalyx. These chains play a crucial role in the cell recognition, helping cells identify one another, supporting immune defense, and determining blood group identity in the ABO system. The bilayer behaves like a liquid crystal sea, with proteins drifting laterally within it. At 37 degrees Celsius, phospholipids stay in constant motion. This is the fluid nature of the membrane. Proteins float within this lipid sea like icebergs, forming a mosaic pattern. This is the Fluid Mosaic Model proposed by Singer and Nicholson in 1972. Watch small oxygen molecules pass directly through the hydrophobic liquid core of the membrane without any help from proteins. Because they are small and non-polar, they dissolve in the fatty acid region and move freely down their concentration gradient. This is simple diffusion. No energy, no protein, no gate required. Glucose molecules, shown as yellow spheres, are too large and too polar to pass through the lipid bilayer. Instead of crossing, they bounce off the membrane and continue drifting until they find a channel protein. The protein changes the shape, opening a passage and allow glucose to move through. This is selective permeability, where the membrane controls what enters and exits the cell. 3 sodium ions, shown in orange, bind to a carrier protein inside the cell. ATP arrives and transfers energy to the protein through phosphorylation, triggering a brief energy flash. The protein changes the shape, opening outward and forcing the sodium ions out of the cell against their concentration gradient. Unlike diffusion, this process requires energy. This is active transport. A large food particle, shown in red, attaches to the outer surface of the membrane. The membrane begins to bend inward around it. This process is called invagination. The indentation deepens into a cup and the edges pinch together, trapping the particle inside a membrane-bound vesicle. The vesicle then detaches and moves into the cytoplasm. This is how large materials enter the cell, through a process requiring energy called endocytosis.